This video covers Chapter 4, Identifying the Causes of Cancer. This is over the second part of the chapter. When we are asking questions like, does a certain chemical cause cancer, we want to, of course, do scientific studies to determine whether that is a true statement or not. And also, in doing these studies, we want to know what dose of that agent would be responsible for causing cancer. So a lot of times we will see a dose-response relationship where if you increase the dose of the reagent, you will oftentimes see an increase in cancer rate. They're often related. And this gives evidence towards cause and effect that the cause of the cancer is due to increasing amounts of the chemical reagent. Here we can see that sometimes there are differences between what a typical human dose is, like what is a human exposed to, compared to what is tested in animals. And oftentimes the dose exposed in humans is very low compared to these higher doses that are studied in cancer. So a lot of times we can extrapolate the fact that we see at these higher doses, we see an increased cancer rate in animals. And then we extrapolate those lines to say, yes, we believe that also this lower dose would have a certain cancer rate associated as well. There's a couple of reasons why this happens. A lot of times humans are exposed to very low doses of chemicals, but sometimes for a sustained time. But in order to actually do those experiments, usually in mice, if the mice were exposed to such a low dose, that the mice just simply do not live long enough to experience cancer. A mouse's life is around 2 years. So in order to actually do these experiments and see the results, oftentimes the higher dose has to be tested in order to actually get any experimental findings. So the next question you might be thinking is it fair that we do these experiments in animals? Does that really translate to what happens in humans? So obviously we have to have some way to test these reagents. We can't necessarily walk up to individuals and ask them to expose themselves to these chemicals so that we can just find out if it causes cancer. So we have to have some other kind of model. So a lot of times we do use a mouse model to answer these kinds of questions. So in this table, it's showing you a list of reagents or chemicals that have been studied in animals and have been found to cause cancer. And so you see the chemical name and then its target, like where does the cancer usually occur, and then kind of like a hint towards the potential mode of action. Like, is it leading to necrosis? Is it leading to hyperplasia? What is kind of like the mechanism of its action there? So we do see some correlation in those animal models, and so we can do those experiments and then extrapolate what we see in those experiments to what we see in humans, and also use that information to make the various rules as far as chemical exposure limits. The Ames test is used as a screening tool to see whether chemicals are carcinogens or not. And it's a unique setup here. So they use bacteria as their method for testing. But they used a modified bacteria. So they use a modified bacteria that actually is lacking the ability to make histidine, which is an amino acid. So since it's not able to make histidine, if it's unable to do that, and it's also growing in a growth medium that lacks histidine, then that bacteria would not be able to grow. So just keep that in mind. That's our model system. Our model system is a bacteria that without histidine added to the growth medium, then it has no ability to grow. And what they do is they're going to take the substance that they're testing and they actually mix it together with liver homogenase. And the liver homogenate will include enzymes that oftentimes if we ingest chemicals into our body, it will travel to the liver. The enzymes in our liver will process that reagent, and oftentimes it's the byproduct of that chemical reaction that results in the carcinogen itself. So this is allowing for that process to happen outside of the body just by mixing it together with those enzymes. They give it time to incubate so that chemical reaction can happen, and then they add it onto the plate that contains the bacteria that is unable to grow without histidine. And then they let it grow. So some chemicals will not have an effect on the bacteria, and you would still see very, very little bacteria growth. But some chemicals will actually have an effect, and you will see that bacteria grow. So you're basically at the end counting how many bacteria are growing in the various chemicals that are being tested. Now, what is happening here is that chemical, if it's able to create mutations in the DNA of the bacteria, that some of those mutations allow for the bacteria to grow in the absence of histidine. So when we see these various bacteria colonies here, these are examples of bacteria that have undergone mutations and are now able to grow in media that lack histidine. So this is a test to show it's like a proxy for saying, yes, there were DNA mutations occurring due to the carcinogen that was added here. So in this case, the higher number of bacteria cells relates to higher ability for that chemical to act as a carcinogen. So in this table here, we see like increasing mutagenic potency on this graph here, and we have various reagents that have been studied. So we have 2-naphthalene, we have the benzidine, we have aflatoxin up here, lots of different examples, and you can see that chemicals will exist on this graph at varying locations because they have different ability to create mutations, and the ability to create more mutations will make that chemical a stronger carcinogen. So in this chapter, we've been talking about epidemiology and how we can use that study or that way of looking at data to understand what are the causes of human cancer. And so the next few slides just kind of go through, and they show you that there are a number of factors that have been shown to be related to human cancer, and it's showing you kind of the data that comes from those studies. Number one on the list is aging. So as we get older, then we have a higher and higher risk of getting cancer. And so what is being shown here in the graphs are colon cancer, lung cancer, prostate cancer, and leukemia. And you can see that with increasing age, that you have a much higher risk of getting colon cancer, lung cancer, and prostate cancer here in these higher age ranges. Here we do see that there is a higher range for leukemia in the older age group, although this graph is slightly skewed because leukemia often occurs in younger patients, and so this graph is a little different because we have a large population of people that get it when they're younger. Number two on our list is environmental and lifestyle factors. So studies have been done to follow various populations of people to see if literally just where they live, whether it's the country, a city, or maybe their lifestyle, their diet, or different various things can affect their rate of cancer. And so what is being shown here are 6 different graphs. So we have 1, 2, 3, 4, 5, 6, and then we've kind of written out what's the summary of each one of these graphs. So for example 1, we have a graph, we have different countries here, and we have kind of like their rate of getting stomach cancer. And we can see that the rate of getting stomach cancer is the highest in China and Japan. And this varies kind of wildly from the United States over here. So there's something about living in China or living in Japan that seems to correlate with having a higher risk of stomach cancer. And there's also studies that show like esophageal cancer is higher there. And sometimes they attribute those factors to possibly, I've heard of them talking about like the fact that they drink extremely, extremely, extremely hot tea. And that is supposed to increase the rates of esophageal and stomach cancer over there. So that's an example of a lifestyle factor that could result in increased rates of cancer. For example, too, it's looking at breast cancer. We see the highest rates are Uruguay and the United States. Example 3 is colorectal cancer, higher in the United States, and that's usually attributed to our diet. Example 4 is liver, and that's the highest in Thailand here. Over here is number 5, we have lung cancer, and it's the highest in Scotland. And then we have prostate cancer, and the highest is in the United States. So some of these factors are attributed to lifestyle. Some of them are attributed to, like the country that they live in, the diet, the types of things that people tend to do in those societies. So that plays a big role. Also, hereditary factors can play a big role. So sometimes people in one country versus another have various hereditary factors. There's various mutations that are passed down within that population that could lead them to be predisposed to various types of cancer. In trying to tease out the difference between environment and hereditary factors, various studies have looked at individuals that live in one country and then move or migrate over to another country. Does that change their cancer rates? So in this study, they're looking at Japanese people. So we're looking at male Japanese people that have a high rate of obtaining stomach cancer. And then looking at those males as they move and migrate over to California. And we can see that they move over to California and their rates actually go down. So it's making you think, like, what is this relationship between where they live and whether they're going to be predisposed to getting stomach cancer? So just looking at stomach cancer alone, you're kind of thinking, well, yeah, I mean, it's the same person or the same group of people. So the hereditary factors should be very similar. So the only thing that really changed drastically here is the environment. So that's kind of leading you to think that maybe it is the environment that plays a big role in their predisposition to getting that cancer. And then looking at colon cancer, so we see that that there's a lower rate of colon cancer in males that live in Japan, but that rate does increase in male Japanese immigrants to California. So it did increase after they moved, right? But then looking here, if we look at native Caucasian males in California, they have a very high rate of cancer. And then we are suspecting that has something to do with the environment that they live in. And so some of these studies look at what is the rate in the country of origin and what is the rate after they've immigrated to other countries. Number 3 on your list is not going to come as a surprise, that cigarette smoking is a major cause of human cancer. So this graph is looking at cigarette consumption over time. So we have, early 1900s over to 2000s, and we can see a sharp increase up around the 1950s, extending up until about the 1975, 1980, and it starts to drop down. And so that cigarette consumption, there was almost no cigarettes being used prior to the 1900s, and it wasn't until after marketing and all of these strategies to promote the product that people started to start smoking, and it became very, very prevalent. And then again, we have more marketing here when we realize, oh my goodness, smoking causes cancer, and then we see that the rates of people using cigarettes goes down. And then looking at male lung cancer deaths, we see that there is a peak in their developing cancer and dying from cancer. We do see that the peak is delayed from cigarette consumption use because we do suppose that there's 10 to 20 or 30-year delay from the use of the cigarettes to actually developing cancer. And so that's why we see that delay in the peak. For females, we see an even further delay because it took a little bit longer for females to catch on to using cigarettes. So they started their peak of using cigarettes a little bit later than males, and so they experience their peak a little bit later here. So lots of studies have correlated cigarette smoking with the development of lung cancer. And it is also correlated with other types of cancer as well, but the one, you're probably most familiar with is obviously them developing lung cancer. But because some of those carcinogens from cigarettes do get moved throughout the body, that it is supposed to have a higher risk of lots of different types of cancer. So an interesting part of this story is that, in the early 1900s, lung cancer was extremely rare. If you were a physician prior to that time, you would maybe see like one lung cancer in your lifetime, if that, of diagnosing patients, right? It was very, very, very rare. But you did see a lot of breast cancer. Breast cancer was very common for a very long time. And it wasn't until after cigarettes kind of came on the scene that lung cancer rates started to rise. And it was pretty obvious to physicians, oh my goodness, we used to never see this. Now we're seeing it all the time. And eventually the lung cancer rates surpassed that of breast cancer. And so that just shows you what a huge correlation cigarette consumption had with lung cancer rates. In these studies here, they've correlated cigarette smoking with the number of cigarettes used per day. So in this graph, we have non-smokers, those that smoked 1 to 14 cigarettes, those that smoked 15 to 24, and those that smoked 25 plus cigarettes per day. And you can see that there's an increased risk of lung cancer according to the number of cigarettes smoked per day. There's also a correlation between the age at which individuals started smoking. You have non-smokers, 25 plus, 20 to 24, 15 to 19, and less than 15. So individuals that started smoking earlier in life have a higher rate of developing lung cancer, suggesting that longer exposure over a long period of time correlates with getting lung cancer. In this study, they're looking at the depth of inhalation. So how deep into the lungs are they inhaling the cigarette smoke? So light, moderate, and deep. So the deeper people inhale, then the higher the risk of cancer that they have. In this study, they're looking at what is the relationship between alcohol use and tobacco use and their rates of developing cancer. So in this graph, they're looking at those two factors. What are they doing to the risk of cancer alone, and then what is the risk of them together? So for alcohol non-drinkers and non-smokers, you see a very low risk. And those that are heavy drinkers but not smokers, then you see a relatively low risk. This is like less than 10. And then here you have non-drinkers, but they are smokers. They have kind of a similar less than 10% risk of developing cancer. Now, if you look at both of them together, heavy drinkers and heavy smokers, they have a huge increase in their risk of developing cancer. And so this increase in the risk is more than if you had added this plus this. Even if we're estimating, we're saying 10 plus 10 would be 20. If that was an additive risk, you would expect the line to be about here. But it's greater than that. And so this is what we call synergistic. That means that the two agents, when they're in there together, that the effect that they have is greater than the sum of what the effect would have been had each agent been acting alone. So it's suggesting that something about the two of them together synergizes to increase the risk of cancer even more. And so kind of an explanation is here. Alcohol can cause tissue damage and cell death. And that process can cause surviving cells to grow and divide. So it triggers proliferation. Tobacco has a different mechanism. It can cause mutations. So when you have those two things occurring side by side, you have something causing mutations, and then you have something that causes tissue damage and cell death, and it causes possibly those mutated cells to survive and to grow and to divide and to proliferate. And that's kind of like a magic situation for developing cancer. Number 4 on our list is red meat, saturated fat, excess calories, and obesity. So things that we eat can cause an increase in the risk of cancer. So the first one is increased meat consumption can correlate with increased colon cancers. So some of this is attributed to red meat. Some of it is attributed to grilling that red meat. And when you grill, that burning and charring that occurs on the meat is a process that can create carcinogens. And so eaten at a high enough rate, this could increase your risk of developing colon cancer. So on a graph here we have meat consumption, grams per person per day, and the colon cancer rate, cases per 100,000. Here's the United States. We're up here pretty high. And then here we have some other countries that eat less red meat than we do, and they fall lower on this graph. So this isn't saying that you should never eat red meat. This is just saying that if you could reduce your daily consumption of red meat, that could help to reduce your risk of developing colon cancer. Other studies have also looked at high fat content, and they show that there is a relationship with high fat and risk of cancer. And just eating higher calories or being obese can also result in a higher risk of cancer. Other studies show that eating fruits and vegetables can decrease your cancer risk. This is due to a couple of reasons. It is leading to just better overall colon health, and so you'd have less inflammation in your colon. And then also these fruits and vegetables carry in them anti-carcinogens, so they have vitamins and all these things that can protect against the development of cancer. Number 5, most radiation causes cancer. So radiation can occur in a couple different forms. One is ultraviolet light. So this is the source is from the sun. We are exposed to ultraviolet lights every day when we go outside. So we have UVA, UVB, UVC rays that come from the sun. In general, UVC is the most, has the highest rate of causing cancer. Although our ozone layer does absorb a lot of the UVC rays, some of them do come through. And so we do have some exposure to UVC. Obviously, our exposure to UVA and B is higher, and they can cause damage, but not as much as UVC. Then we have ionizing radiation. This is a much more dangerous type of radiation. It's a little bit stronger than UV. And we would get that source from X-rays and those kinds of imaging things. And so that can increase our risk of getting cancer, and it can actually penetrate into our body further. So we have a higher risk of cancer in our internal organs. So we have a chapter that talks about radiation, and we'll get into the kind of concept of radiation. But it is high energy electrons that are coming in, and then when they interact with the cell, that they can cause damage on DNA itself. Number 6 is viruses. So there is evidence that there are oncogenic viruses. That means a virus that can cause cancer. There are 3 examples given here. Epstein-Barr virus can cause Burkitt's lymphoma. Human papillomavirus, HPV, can cause cervical cancer. Hepatitis B and hepatitis C can cause liver cancer. So these are known facts. Many studies linking these viruses with the ability to cause cancer. And again, we will explore these in more detail in another chapter. On the right, you see a figure that is showing you kind of the basics for an experiment that was done a really long time ago. So I don't remember the year, but I believe it was in the 1800s. So we have a guy named Peyton Rouse, and he did an experiment because he had experience with chickens that were developing sarcoma. So it was kind of like common, like, yes, these chickens can develop cancer, and it seems to be infectious, because if you have one chicken with it, then pretty soon you have 5, and pretty soon you have 10 chickens with it. So it really seemed like it spread through the population. So in this experiment, they're investigating, is it possible for whatever is causing these cancers to be transmitted through the fluids in a chicken. Because back then they didn't have a lot of information about viruses, bacteria, stuff like that. So what they did is they took that tumor out of the chicken. So they've taken it out from the chicken, they grind it, they run it through a filter so that they're only leaving like the liquid part of this. So this would not contain the cells from the tumor. It's only just kind of like the juicy, liquidy part. So cell-free extract. And then they're injecting it into a healthy chicken to see, is there some property, not the cell, but something else that can transmit the ability to develop this tumor. So they inject it into a healthy tumor and they developed a sarcoma. So we will talk more about this as well because this study led to the idea that something smaller than a cell could cause cancer, and also further studies led to studying the type of virus that it was, but also the gene that it encoded, that is a well-known oncogene now. So in the last few slides, we've been looking at the top leading causes of cancer deaths, and we've kind of just run through the biggest list. We haven't talked about every cause known to mankind, but we've kind of hit the big ones. We've obviously talked about age, which is not on here because it's not like really a factor. It's just part of life. And then we talked about tobacco, diet, infectious agents, ionizing radiation, alcohol, UV. Some other things that we didn't really talk about. Hormones is known to cause cancer, some occupational exposure to various things, pollution, drugs, additives, and there's a big group of unknowns. So this is not an exhaustive list of things that can cause cancer. Anything that can cause a mutation or constant inflammation has the ability to cause cancer, but we've kind of hit the major ones in this chapter. So that wraps up this chapter. Thank you for listening.